Hi everyone, today we'll be going through A-Level Biology AQA Paper 1 Predicted Paper for 2024. Please still revise every topic since there's no guarantee what will come up on the day. I have timestamps in the description so you can go over a certain topic if you want, and I have put all the predicted topics here and wasn't able to include every topic in this video, so I have linked a second video in the description that goes over the other topics that may have come up. Don't forget to like and subscribe and turn on the notification bell. Let's get started. Question 1. Species richness and index of diversity can be used to measure biodiversity within a community. A. What is the difference between these two measures of biodiversity? We can do this by just listing the definition of species richness and index diversity. So we know that species richness measures only a number of different species, and it does not measure the number of individuals, which is what an index of diversity does. Scientists investigated biodiversity of butterflies in a rainforest. Their investigation lasted several months. The scientists set one canopy trap and one understory trap at 5 sites. The canopy traps were set among the leaves of 16 to 27 meters above ground level, and the understory traps were set under trees at 1 to 1.5 meters above ground level. The scientists recorded the numbers of each species of butterfly caught in these traps, and the table below summarizes their results. In the traps in the canopies were set at 16 to 27 meters above ground level. Suggest why there was such great variation in height of the trap, and this is simply because trees varies in height and you want to make sure that you get every single type. C. By how many times is the species diversity in a canopy greater than its understory? Show your working and use the following formula to calculate species diversity. This is where N is the total number of organisms of all species and N is the total number of organisms in each species. First we have to do this for the canopy, and we also have to do this for the understory. So let's start with the canopy. First thing we do is that we add all of these together. Overall, there is 196. So this is what the first one means. It means 196, and then we do 196 minus 1. So we times it by 195. Now we divide it by, this is the sum of each individual one. We do the same. So let's say for this one, it'll be 15 times by 15 minus 1, so 14. Here it'll be 14 times 13. Here it'll be 25 times by 24. 89 times by 88, 21 times by 20, and 32 times by 31. And once you do all of these, again, you add them up all together. And if you add them all together, you are going to get 10,236. Now we divide both of them together, and if you do that, you will get 3.73 as its index of diversity for the canopy. Now we do the exact same with the understory. We do the exact same method, and if we do that, you will get 3.30. I recommend that you try it out and pause the video where the table was so that you make sure that you understand how you do this index of diversity. Because they're asking for how many times greater the canopy is, we just have to divide them by each other. So we do 3.73, and this is over 3.30. We put the canopy first, and if you do that, our answer will be 1.13 times bigger, and that is our answer. The scientists carried out a statistical test to see if the difference in the distribution of each species between the canopy and understory were due to chance. The p-values were obtained and shown in the table and explain the results of the statistical test shown. We can see the p-values for all of this. We can see for most of them except one that the p-value is less than 0.001. However, for one of them, it is more than 0.05, and that is where first thing we are going to talk about, because that is the anomaly. So what we can say for this species, we can say that the difference in distribution is probably due to chance, and it is due to chance because the probability, the p-value is more than 0.05, meaning it's more than 5%. Therefore, we say it's due to chance. However, because all the other ones are p less than 0.001, we can say this is very highly significant, much lower than 5%. So its difference in distribution is highly unlikely to be due to chance and actually due to something else, such as distribution of trees. Describe and explain how a counter-current system leads to efficient gas exchange across the gills of fish and water. So what it is that, let's say this over here is the fish and this over here is the water. So while the fish, their blood goes this way, while water goes this way. This means that because they flow in opposite direction, there is a concentration gradient always. So while it's high over here, it's low over here. And where it's low over here, it's high over here, which means that there's always this concentration gradient where they're able to go and they're able to exchange resources that they need. Let's say in comparison, if they were both going the same direction, like over here, they're both going the same direction, this will both be high concentration in both ways, and there will be still low concentration in both ways, which means after a while, their concentrations will be the exact same, so there's no more movement and gas exchange between them, which of course is inefficient because they want to get as much resources as possible. So them having different concentration at different time is the reason why gas exchange is so efficient. And this happens across the whole gill, not just parts of it. So B. Big gill disease is caused by a parasite that lives in gills of some species of fish. So the disease causes the lamellae to become thicker, this is important, and to fuse together. So it reduces efficiency on gas exchange, and fish give two reasons why. So while they're talking about fish, this is basically the same. They have the same efficiency, same reasons why they're so efficient as a human does, as any living organism does, and this is when it has larger surface area, and when it's thinner, we have more efficient. That's the same with our alveoli. So we just apply the exact same principles. So it says over here that becomes thicker, so this means there's longer diffusion distance, which of course makes it less efficient. And it also says that it fuses together. Because it fuses together, that means there's reduced surface area. So always make sure that you remember these two reasons and try to use it in the application that they gave you. These type of questions just wants to test how you apply your already given knowledge into a question. So even if you see a question that you completely don't understand, you do know the answer, you just need to find a way to know your existing knowledge into what the question really asks you to do. So C, the table below shows some feature of gas exchange of a fish at rest. So calculate the volume that would have to pass over gills each hour to supply the oxygen required by fish. So show your working. So what this is that you basically do 0.4 times 90, do the oxygen required for fish per kilogram. So that means we need to times it by the mass. This will equal 36. And this is the volume absorbed of each water centimeters. That means you divide it by 7, and which is 5.14, which is the answer. Always try to put it at least into 2 to 3 significant figures, 2 decimal point places. So don't just round it to the whole number. So the volume of water passing over the gill increases if the temperature of the water increases. So suggest why. This is mainly due to increased metabolism. When the temperature increases, there's increased metabolism, increased respiration, also increased enzyme activity. This is generally because of increased kinetic energy within the water. Question 3. Explain the features of a gas exchange surface that are common in both insects and mammals. First, we can say large surface area and short distance for diffusion. These are very common for every type of gas exchange surface. It's the principle of how they work. So all we have to do is just link how an insect and a mammal have this large surface area. So we can say large surface area. This is because in mammals we have lots of alveoles, and in insects we have lots of tracheoles. We can say it has a short distance for diffusion. This is because of the alveoli. Again, it has just one cell thick, and as well as the tracheoles, both have thin walls, and also it's moist, and this allows gases to dissolve. Another one can also be that tracheoles penetrate tissues, and alveoles are also close to a blood capillary. That means they maintain concentration gradients. 4. Scientists investigated the transpiration of a pine tree over a 24-hour period. The mass of water lost per second and the total area of leaves were measured. The decrease in diameter of the xylem vessels were also measured in the same 24 hours. The results are shown in the graph. Use your knowledge of cohesion tension theory and explain the changes in xylem diameter and transpiration rate over this 24 hours. if we just look at the graph and we'll see whatever trends that we can see in the beginning, during the night, which is in these times over here, we can see that the xylem diameter and the transpiration are both really low. They do not change. However, during the time it increases a peak between 12 and 1 o'clock, and then it decreases once again when it turns to night. That is the first pattern we see. And with graphs, the first thing you want to do is mention whatever the obvious is, whatever the pattern of a graph is, that will usually snatch you a couple of marks. So what we can say that the rate of transpiration increases in the day, and the fluctuation in rate of transpiration, what's the reason? It could be due to environmental changes such as wind speed, rain, humidity, cloud cover, or even dehydration. And we can say this change in the pattern of xylem diameters, they follow the same trends as the change in transpiration. We can see that both of them are very closely linked together. They are together. Now, what's the reason for this? What's the reason of the fluctuation? What's the reason of increase, the change, the not much change during the night? This could be because the light causes the stomata to open, and also increase in temperature because of increase in light increases this kinetic energy. Also, reduced humidity can also increase concentration gradient, while increased wind speed increases and it maintains this concentration gradient. This is all reasons for why there's more transpiration during the day. And also, evaporation from the leaves reduces the water potential in the leaves, therefore more transpiration. Now, linking back to cohesion tension theory, because that is exactly what they want you to do. You would not get all the 6 marks unless you do exactly what they tell you that they want in the question. This could be because water molecules are polar, so cohesion is due to the hydrogen bonds between this water molecule, and columns of water is under tension as this water evaporates, so evaporation causes the pressure to decrease, and therefore narrowing this xylem, and that's why it becomes smaller. 5. Enzymes are involved in the breakdown of carbohydrates. The diagram shows structure of sucrose. The enzyme sucrase breaks down the sucrose into 2 monosaccharides. Which type of reaction does the sucrase catalyze? So they say that it breaks down, and breaking down is a hydrolysis reaction. 2i, name the monosaccharides produced from this breakdown of sucrose. We can say that these 2 sucrose, all we need to do is just identify what these type is, and you do need to memorize what 2 monosaccharides make up the disaccharide sucrase, and this is glucose and fructose. The diagram shows one of these monosaccharides. Draw the structure of the other monosaccharide. So we can see over here that they have already drawn this first one. So all we have to draw is the next one. So we already know that this part is already given for us. So if we just continue and we just draw this. Now all we need to care about is what's going to happen over here because of this hydrolysis reaction. With this hydrolysis reaction, what it does is that it adds H2O. So if you see over here, you can see that it added an H and another H to that O over here. So what is left over is AOH. Question 6. Enzyme TK is found in a human cell, and TK exists in a non-functional and a functional form. The functional form of TK is only produced when a phosphate group is added to this TK. As shown over here, only when this phosphate group comes, then you can see over here that this active site changes shape so that it can actually go into the substrate. So addition of this phosphate group to the non-functional form of TK leads to the production of the functional form of TK. Explain how. So as we can see over here, phosphate group, once attached, it changes this shape and changes the shape of the enzyme, which means it changes this active site. Once it changes this active site, it can actually bind to the substrate of TK because the active site forms and becomes the right shape. So then again, the golden keywords for enzymes is that it can form E to S complexes. B. The binding of functional form of TK, its substrate level, leads to cell division. Chronic myeloid leukemia is a cancer caused by faulty form of this TK. Cancer involves uncontrolled cell division. So figure 2 shows the faulty form of TK. You can see over here that this part over here is already the active site that we need type of shape. So just how this faulty TK leads to a chronic myeloid leukemia. So you can see it already has a functional active site without the need to attach this phosphate group. And because of this, it means that it's functional all the time, so it's always dividing. Therefore, it cannot be stopped. It's not controlled by phosphate. C. Imidazole is a drug and it is used to treat chronic leukemia. Show how this drug inhibits the faulty TK and use all the information and describe how this stops the development of the chronic leukemia. We can see over here that this is the drug they talk about. This, once it forms, you can see that this changes the active site, so it can no longer bind to our substrate because the active site has changed, the normal ATS complex can be formed. What type of inhibitor is this? This is a non-competitive inhibitor, which means that it binds to the site, any other site in the enzyme other than this active site, and this causes it to change shape and change the shape of these active sites. So it causes TK to be in its non-functional form. So the active site is no longer formed and E to S complexes cannot be formed. So uncontrolled cell division is now stopped and more controlled. Question 7. The figure below summarizes the way in which cheese is made. We see we have fresh milk and then we add chymosin and then we get milk and it's separated by draining. So we get two types. We get whey and curd is the thing that makes the mature cheese. So A student carried out a burette test at the sample of whey. The sample turned purple and explain why using the flowchart. So this chymosin is actually an enzyme, and because it's an enzyme, all enzymes are proteins, so therefore that's why it turns purple, because it turns purple. Biodiversity test turns purple if they see this peptide bonds present, which is present in any type of protein, which an enzyme is. B. The percentage of protein in mature cheese is greater than the percentage of protein in milk. So use the flowchart to explain why. This is because, as we can see here, it's separated by draining. That means draining just means that they remove the water, and that's why there's a greater percentage. Question 8. A frog heart was attached to an instrument which measured the force produced as the heart is contracted. Graph 1 shows the changes in force when the heart was bathed in a solution of salts at 20 Celsius. Graph 2 shows the result of heart that was bathed in the same solution at the same temperature, but includes acetylcholine. Point A and B shows when the atria and ventricles were contracting. Which point A or B shows the contraction of the ventricle, and give 2 reasons why. So you can see over here that it is B, and this is because, first of all, the atria is the one that actually it contracts first, it's the first thing that goes through, and then the ventricle second. So the fact that it occurs after just shows already that it is the ventricle, and also because it happens at such a more higher force. If you can see over here, the force is completely different, much higher, and this is because it has to pump to the rest of the body. B. Calculate the frog heart rate when acetylcholine was not present and show your working. So graph 1 is the one that is with no acetylcholine. What we do is we see how much it is in one cycle. So our cycle can be from here to here, and we can see that is around 1.6 seconds in each one. So we do 60, divide this by 1.6. If we do 60 divided by 1.6, this is the amount it takes for one cycle to occur. We get 37.5, which is our answer, 37.5 beats per minute. From the graph, what can you conclude about the effect of acetylcholine on the heart rate? If you see our heart rate is how many times the cycle occurs. We can say that over here there's much more than over here. The cycle goes 3 times over here, while here goes 5. So we can say it's reduced. On the stroke volume, we can see that the forces are basically the exact same, which is the amount of pressure that the blood goes into, which is our stroke volume. Because it's the same, we just say it's no effect. Use your answer to part I and explain the effect on acetylcholine on the cardiac output. This is reduced because of the heart rate being decreased, and we know that our equation is cardiac output, and it is how we get cardiac output. We do heart rate times by stroke volume. Because heart rate decreases, that means overall our cardiac output will decrease. Addition of acetylcholine in the experiment mimics the effects of one branch of the autonomic nervous system. What branch is this? Because it's autonomic, this is the parasympathetic. Explain how nervous control in a human can cause this increased cardiac output during exercise. This is because it's coordination via the medulla, which is in the brain. This is the cardiac center. So this increases impulses along the sympathetic nerve, and this is to the SA node, which is the pacemaker. This means that more impulses are sent and increase the discharge of the SA node, which is the pacemakers. Explain why increased cardiac output is an advantage during exercise. Everything to do with exercise, we always just want more energy. So what we say is that in exercise, more energy is released, and this is always for aerobic respirations to help the muscles, because of higher cardiac output will always increase in oxygen supply, glucose, increases CO2 removal, and lactate removal. So it all helps for respiration, and this is always to and from the muscles. Question 9. A student investigated the effect of temperature on the release of pigments from the pieces of the beetroot. She cut a fresh beetroot in 4 pieces and placed each piece into a water with different temperatures. At 10 minutes, she removed the beetroot and used a colorimeter to test how much pigment has entered the water. She placed the colored water into the colorimeter and measured the percentage of transmission of light through the water, and her results are shown in table 2.1. Here's the temperature, and here's the percentage of transmission light. The results show that below 50 Celsius, little pigment has entered the water. Explain why there was no transmission light after the beetroot has been placed in 100 Celsius. So at 100 Celsius, the membrane is completely damaged. The cell surface is completely damaged because it's too much of a high temperature. This means that percentage completely leaks out, so it absorbs and completely takes up all the light. So just 3 ways in which the student could have improved her investigation. So if you just look at her method, we can just see whatever she didn't include. She didn't include anything to do with volume. She didn't include anything to do with size of beetroot, what type of beetroot. So anything that she has missed that you usually need to have in a method, equipment, volume, size, concentration, anything to do with that, you can put as a improvement as they missed it out. So what we can say that we want, first of all, we can say we need a fixed or the same volume of water for each one, all the same sample size, surface area. We can also say cutting to increase the surface area, all the same size and width. We can say also that same beetroot is used, or at least same part of the beetroot, since some parts might have more pigments than the other. We also can say that we want more samples at each temperature. The more samples we have, the more we can see if something was an anomalous result, so we get a better mean. We can also say we want blotting and rinsing after the cutting, so we remove any pigment that was already there from the cutting, and more intermediate temperatures for more results. 10. Why is the genetic code described as being universal? This is because in all organisms, they all have the same DNA with the same triplet codes that all code for the same amino acid in every type of organisms. The genetic code uses 4 different DNA bases. What is the maximum number of different DNA triplets that can be made using these 4 bases? All we do, because we know that there is 3 different types in every single codon in a DNA triplet, so we do 4 times 4 times 4. We just times it to each other, and our answer will be 64. Transcription of a gene produces pre-mRNA. Name the process that removed bases sequence from pre-RNA to form mRNA. This is called splicing. C. The figure below shows part of a pre-mRNA molecule, and geneticists identified 2 mutations that can affect this pre-mRNA, as shown in the figure. We have base sequence coding for amino acid. This is the exon. We have intron, exon. Base removed from pre-mRNA, and again, base sequence coding for amino acid. So we have mutation 1, which is deletion, and mutation 2, which is a substitution. Mutation 1 leads to the production of a non-functional protein. Explain why. This creates because the mutation completely changes the triplet, because this is a deletion, and because this is a deletion, it causes this frame shift. So this changes the amino acid sequence, so it codes for all different amino acids because the triplets have changed. If different amino acid sequence, that means it affects hydrogen, ionic, and sulfur, and all those type of bonds, which is our secondary structure. Changing secondary structure, therefore, also changes the tertiary structure of a protein. Therefore, it becomes non-functional. If they did ask about mutation 2, this will not have any effect because the only ones that will have effect is on the exons. In an intron, it won't have effect because either way, it's completely removed from pre-mRNA. Question 11. Changes to protein code of the influenza virus causes antigenic variability. Explain how antigenic variability has caused some people to become unaffected more than once with influenza viruses. This is simply because memory and B cells, which is the things that allow us to remember when we were infected with a certain thing, does not recognize these new antigens that have presented. This is because antibodies previously produced were not effective at all, as the shape is not complementary with this new antigen. B. The drawings show changes in B lymphocytes after stimulation by a specific antigen. Describe the role of the macrophages in stimulating B lymphocytes. What they do is just antigen in the membranes are presented to the lymphocyte. This produces cytokinesis. 2I. Explain how changes is shown in the drawings are related to the function of B lymphocytes. If you look into the drawings, what we can see is we can see an increase in Golgi apparatus. Here we see 1 mitochondria, while here we see much more. And we also see an increase in ribosomes. So these are the 3 things we are talking about. This is sometimes as simple as just cell structure. Just see any of the difference, and that's what we're going to talk about. So what we can say the mitochondria, more mitochondria means that more ATP, more energy. We can say more ribosomes or more RER can synthesize more proteins, which of course is good. We have Golgi body and we have more of those, so it secretes, modifies, and packages proteins, and also produces glycoproteins so that they can be used to see and mark new antigens. And also B lymphocytes, they produce antibodies. That's why at the end of the day, they're all related to the function of B lymphocytes. An antibody here are proteins. 12. The epithelial cell in the line of the small intestine are adapted to the absorption of glucose. Explain how. So first we are going to talk about, when we say what's adapted, we talk about features. We talk about the most simple things, which are cell structures and features. So that's the first thing we are going to do, and then we talk about more about its function. First thing we can say is that microvilli, this produces a large surface area. We also have many mitochondria. This produces a lot of ATP and provides energy for any active transport that may be needed. And they also have a lot of carrier proteins, which is good for active transport, which they always do for the absorption of glucose. Also lots of channel and carrier proteins. This is for facilitated diffusion, because facilitated diffusion always happens before they need to use active transport and energy. They also have co-transport of sodium ions and glucose, and they also have membrane-bound enzymes, so this can digest disaccharides so that they can produce monosaccharides and produce this glucose. Question 13. When a vaccine is given to a person, it leads to the production of antibodies against a disease-causing organism. Describe how. This is because a vaccine contains this antigen from the pathogens, and macrophages are then presented and the antigens on its surface. T cells then become complementary with this receptor protein will bind to these antigens, and T cells later stimulate the B cells with complementary antibodies on its surface. So then B cells are going to secrete a large amount of antibodies, and also B cells does divide to form cloning, all secreting and producing the same antibody once they figure out which one, and this all helps to go against a disease-causing organism. Question 14. Explain 5 properties that make water important for organism. First of all, it is a metabolite for different reactions. This includes condensation, hydrolysis, photosynthesis, and respiration. It is also a solvent, so it allows metabolic reactions to occur, as well as allowing transport of substances. It has a high specific heat capacity, and this means it buffers changes in the temperature. Therefore, it can be a really good environment such as the water, so it doesn't change temperature easily, so it is good, stable environment for organism living there. It also has a large latent heat of vaporization. This means it provides a cooling effect through evaporation, so let's say sweating helps us cool down. And also it has cohesion, which is between the water molecules. This can be to support columns of water in plants, such as the transpiration stream, and also produce surface tension in supporting small organisms. Thank you for watching. If you have any questions, whether it's to do with this predicted paper or content in general, please let me know in the comments. I'll be happy to help. And good luck for your exam. I hope it goes well.